आफूले भुल्न भनेको त हो। Hello, everybody. Hello, Daniel Than. Hello, Daniel Than. Can you listen me? Hello, Daniel Than. Hello. Hello, Alini.
Hear you. How are you?
Yeah, I'm very good. Can you listen me?
Yes, of course.
Yeah, fine. Okay. Hello, Professor Than. Professor Than. Hello, Dr. Manzoor Saab.
Hello.
Can you listen me? Okay, fine.
Yes,
Okay.
yes, I can listen you.
Hello, Professor Daniel Than. Can you give? I'm just waiting of your response. Then we will start after 8 or 9 minutes, 10 minutes.
Okay.
Okay.
Hello,
okay.
Professor Than. Hello, Professor Than. Hello, Professor Than. We are just waiting of you. Then we'll start it. So I hope everybody can listen my.
You can listen me clearly?
Yes. Your voice is very clear, sir.
Okay.
Yes.
Hello, Professor Than. Professor Than, please unmute your mic. Yes. Hello, Professor Than. Please make your unmute your mic, please.
Yeah, I'm remute the mic. I think you can hear me.
Yeah, it's good. So everybody's there now. Okay.
Just quickly share my screen. Just make sure that you can see everything. Let's make sure. Remember how to share. There it is.
Yeah, please.
You can see?
Yes. Yeah, we can see. Yeah,
Okay.
we can see.
Good.
Yes.
Okay,
Yeah.
I'll
It's fantastic. Yes. Dr. Manzoor, can you see this PPT from Professor Than?
I'll unshare. I think you can see. Yeah?
Yeah.
Okay,
So,
unshare.
Sir, Dr. Manzoor, can you see the slides from Professor Than?
Okay. Yes, I can see.
Okay, Than, it's working. Yeah, it means everybody can see. Just please wait. We will start after 5 minutes.
Okay,
Do you want to try to share my screen also?
Yeah. Why not? Please share your screen as well.
No. Can you see it now?
No.
Just a minute.
Yeah. So there is a link next to leave. So you press.
Yes, I can see it. But I don't know why.
You first open your PPT then share.
Yes,
from-
I have it open. I have
Oh,
it
okay.
open, my PPT, and I went to
We did the rehearsal last time, so it was working with you.
yes, we have some problem, but I will try. If you we have time, I will try once again, and then
Leni, at least there should not be problem today.
okay. Yes, you have my presentation.
Yeah, I have your presentation, but try to share yours.
Yes, but I don't know why. No, I cannot.
Oh,
It's don't work only okay, just a minute.
yeah.
You can see this.
No.
You can see this.
Nope.
No, again,
Oh.
because I can share it, but it's only to you that this happened. Now?
No.
Not now. Okay.
Yeah.
Then Professor Azar, you will have to have my presentation. Okay.
Oh, okay. Okay. Let me show your PPT if it is fine with you. Okay.
Yes.
Yeah. Can you see this?
No.
Oh, I share this right now.
Yes, I can see.
Leni, I shared, and Dr. Mazur, he can see.
But me, I cannot. Why? I don't understand.
I don't know what's the issue with your system. But
Because
Dr. Mazur
I
can
can see
see
Professor Tan. I can see Professor Tan presentation, but not yours
Then there is an
or
issue
mine.
with your. Oh.
I cannot
So
see
please see.
your
There is a
present. I
problem
cannot
with your
see.
system.
Yes, but I can see the other presentation and not yours or mine.
I think there's no problem. What you can do is just share your screen, and then after that, you can tell her what slide it is. Once she knows what slide it is, she can talk. So it should be fine.
Actually, she's not able to see slide shared by me.
That is okay, because you can show the slide and you can tell her what slide number it is. She can use the same slide number.
Yeah.
You'll be fine.
We will try. We will try this.
Yeah,
Okay.
you open slide. Alini, you open your slide, just see, and then when you want the next slide, let me know. I will shift.
Okay. Okay.
Yeah, that would be fine. Okay, friends, colleagues. Now we are going to start today's session. So everybody, good afternoon, good evening, and some there's good afternoon. Good afternoon, and in and is also afternoon. So today we are having our webinar. The title of webinar is so this is the first webinar from our COT research group, and so here we are having our professor, Dr. Zulfikar Ali. So I request Professor Dr. Zulfikar Ali for some keynote and some opening remarks. Then we will proceed for the presentation from Professor Tan. So over to Professor Zulfikar. Hello, sir.
Yes, thank you very much, Dr. Tessin. Am I audible?
Yes, yes.
Yeah. So thank you very much. Good afternoon and maybe good morning and good evening as well. The people they are sitting from different you can say geographical areas. So thank you very much. Especially I will welcome Professor Khan, Alini, and Manzoor Hussain, who are the presenter of today's webinar. So thank you very much and welcome in this webinar. In Pakistan, cotton basically over the years its cultivation was decreased, and particularly if we see the cotton cultivation, so there are three major factors which are impacting its cultivation: the production practices and the production or overall the yield. And second is the climatic factors. Although this cotton plant is a desert, we say it a desert plant, but it is very much prone to the climatic changes and the climatic changes, particularly impacted by the insect pest and the heat waves. So the third factor which impacts the cotton cultivation is the marketing. So these are the main three you can say the driving factors of the cotton cultivation. So here in Pakistan, this production is very much challenging, and the farmers they reduce their cultivated areas of the cotton. So today's webinar is very rightly will address this sustainability of cotton production, particularly and the pre-breeding of heat tolerance. These will be the knowledge, or you can say the experiences we will learn from the speakers how to revive this cotton cultivation in Pakistan, because Pakistan is agriculture country, rural economy, and in the rural development as we say, so Pakistani rural women, they are very much involved in the cotton production process, particularly in picking cotton, because in Pakistan the hand picking is there, and in the production practices, in the farm operations, several farm operations, the females they are involved. So in the rural economy, where this crop is very much related to the prosperity of these this segment, and particular the female segment. So it is very important here in Pakistan to not only sustain the cotton production, but also enhance the cotton production as the population is growing. So I think we will enjoy the experience of the learned speakers for this sustainability and particularly addressing the climatic changes. So thank you very much once again, and welcome to the speakers to this seminar. So thank you.
Okay, thank you very much, sir. Thank you very much. Okay, our first speaker is Professor Daniel Tan. So I want to share some biodata of Professor Tan. Professor Tan is full professor and leading the high temperature crop physiology laboratory at the University of Sydney. So he has led heat tolerance projects in cotton, chickpea, wheat since 2005, and he has supervised over 13 PhD students currently completing the work on high temperature and abiotic stresses in cotton. So today, Professor Tan is going to present his work about the pre-breeding for heat tolerance cotton. So over to Professor Tan, please, Professor.
Okay, thank you very much. Can you see my screen? Maybe not yet.
Yes, yes, we can
You
see.
can? Okay, good, good.
Yeah.
Okay, I'm very pleased. Thank you very much for inviting me to give this talk. Yeah, it's a real pleasure. One day I'll come and visit Pakistan because I have many students from Pakistan and I love the country. So this first talk that I'm giving is a collaboration. So a lot of my work is funded by the Australian government, the Cotton Research and Development Corporation, and this work is also a collaboration with CSIRO, the Australia's science organization, and also Texas A&M in the United States, where we'd have a collaboration. Okay, so this is the outline of the seminar. So we'll just quickly go through the Australian cropping environment, which is quite similar to Pakistan. Maybe we're not as hot as in Pakistan, some areas. And we'll discuss the projected global warming, the cotton breeding in Australia, and then we'll talk about the heat tolerance work that we've been doing over the last 20 years. So if you look in Australia, you can see this is a sort of a spatial temporal map of the rainfall. The blue is where we have floods. Like now we're having floods now because it's La Nina stage phase. But in the last few years, you remember like 2 or 3 years ago, 2018, 2019 and before, we had droughts. You can see already red. So we either have a drought and it's very hot and dry, or we have a flood where it's wet and cold and wet. So we have this sort of cycles where we have some flood and then some drought. So that's why we have to not just look at heat tolerance, drought tolerance, and also flooding tolerance. So it's quite complicated. So we know from global warming, the estimated temperatures are projected to increase. And in Australia, all the projections are showing that we'll get fewer frosts and more high temperature days. So before I go on, I'll quickly discuss about the cotton breeding program we have in Australia. A lot of the cotton breeding is done in a collaboration between CSIRO and cotton seed distributors. This is like a cooperative that produces the cotton. So next time when you come here, I think you have sent a few delegations down to Australia before. We will take you out to Narrabri, our campus there, and you can see the operation with the breeding in partnership with the seed production. So this is our Cotton Research Institute. So we have two institutes here. We have the Cotton Research Institute, and then further down the road, down there, we have the Wheat Research Institute. Wheat Research Institute is run by the University of Sydney, and the cotton one is run by CSIRO and New South Wales Department of Primary Industries. But we have very close collaborations. So you can see where do we grow cotton? So we grow cotton in most areas where we grow wheat, but where the rivers are. So most of our cotton is irrigated, as you can see here. So Narrabri is right in the middle. Okay, and Sydney is here. So you can see most of our cotton growing areas is the eastern part of Australia, but we're slowly expanding it further north. We used to grow a lot of cotton up north as well, but we had problems with insect problems. But now, because we have genetically modified cotton, we can start growing it up north as well. And they're going to build 2 more gins up north as well, so that to support the industry up north. So northern Australia is very similar to the climate in Pakistan. Okay, so this is the process. This is just published, I think last week. My colleague, actually not my colleague, my colleague now, but formerly Warren Connerty was my PhD student. He's now one of the breeders there in CSIRO. So what we have is we have a combination of conventional breeding and GM cultivar development. So first of all, we do the common pedigree method. We cross them, and we self it, and then we also test them in different environments, right? We have a hot environment in the northern part of Australia, middle, and then the southern part of Australia. And we also, after we get the varieties that we want from the conventional breeding, we will back cross it to a donor, a GM donor, genetically modified, basically a coca that has been a coca variety that has been transformed with all the genes like the BORGAT3 genes, the Roundup Ready flex genes, and we will back cross it. Oh, maybe I got it too fast. Back cross it until we get advanced lines, and then we'll test it in the field as well. And then we will seed increase. We pass it to cotton seed distributors, and they will seed increase it. Usually with farmers, right? Good seed producers, they will seed increase with farmers that are expert in producing seeds. Okay, so it's very conventional. But what happens is that most of the yield increase is due to our conventional breeding, because as you all know, heat tolerance and yield are all to do with polygenic traits, whereas the GM traits are mainly protective, like, for example, BORGAT is protected from insects and maybe from herbicide damage, right? Okay, let's have a look. So we look at this is just a simple diagram. What happens is that over the years we have different varieties that are released. Let's say Sacra is released in 1986, and then Seocra 1998 was released then. And what we did was we planted them, from 1980 to 94. The same varieties, all varieties, we plant them all together, and we found that there is an increase in yield. But when we planted later on, the increase was even higher. Look at the slope. And that's because it's not just the varieties that are resulting in an increase in yield, but also our practices. So it's an interaction between the variety and the management practice. Okay, so you can see that in this is all in that paper, that the Connerty paper as well. About half of the yield increase over time is due to cultivar development, and about 1/4 due to management, and a quarter also, this is a secret one, a quarter is due to the interaction between cultivar and management. So we have better management, better cultivars, and that's why we got very, very high yields, not just cultivar development, but an interaction between the two. So you can see our cultivar, this is the old days, conventional cotton days. You can see that our cotton varieties are very dynamic. We don't keep our varieties for long. It goes, this is, it goes up, there's a lot of adoption, and then after that, it dies down, and then the next variety comes in, next variety, and the next variety. And now the GMO varieties are also the similar pattern, right? So these are all the conventional varieties in the old days, just to show the change over time. Okay, so you can see now from 1960 to 2020 that we have been quite fortunate in the yield increase. We have got like 33 kilograms per hectare increase per year, right? So it's quite a good yield increase over time, and we are, at the moment we are one of the highest yielding in the world. I think it is the highest yielding in the world. Maybe Israel beats us by a bit, but we have, commercially we got the highest yielding cotton, and we're not using hybrids. I went to India last time in the conference, the one WRC, I think CLC 5 or something. And the Indians also have, I mean, their yields are lower, but they have hybrids. All our cotton are open pollinated varieties, no hybrids, right? Because we want small plants that we can mechanically harvest. So fairly high rates, as you can see, the half is due to the conventional breeding, and then there's others due to the interaction between better management and breeding. Okay, now let's get to heat tolerance. We started the heat tolerance work with CSIRO with my first PhD student. She was actually my first one about 3 years back now. And it was a collaboration with CSIRO and Texas A&M. And some of the techniques that we developed are still being used today. So what we did was we looked at different levels, similar to Pakistan, because I've marked many Pakistani or Faisalabad thesis that use a similar method. We look at it from the crop plant on a field level, looking at yield, fruit retention, fiber quality, and also at a leaf level, because we want to look at photosynthesis, gas exchange, transpiration, fluorescence. We do it both in the field and the growth cabinet, and we do it at a cellular level, looking at enzyme viability and membrane integrity in the field, growth cabinet, and laboratory. And we also look at gene expression, mainly in the growth cabinet. So let's have a look at some of the data. What we did was, this is the old days. We now have actually better technology. We have actually got chambers that in the field that we have special heating. But at the time we first started this project, we just use tents. I think we have seen a photo of it like this. So it's passive heating from the solar radiation. No air conditioners or whatever that we use nowadays. So very simple passive heating. And you can see the yields are much lower when we have the tents on. And we use electron transport rate, which is like a photosynthesis, electron transport type, and we can get a good correlation there. So we're still using this method at the moment. And we also use enzyme viability. This is using tetrazolium, very simple test, tetrazolium test. And if it's viable at the control, it turns it to a rate formazine. And if it's dead, it's not viable at like 45 or 90 degrees, it'll just be green. The chlorophyll just shows there's no life there. And it's been developed further, and I think Jaconis, she was working with us for a while, but now she's in cotton. I think the U.S. cotton incorporated in U.S. now, working there. But she further developed this method. And here we looked at membrane integrity, which is like basically just electrical conductivity. In a hot year, those tents work really well. You can see that's good relationship. But I mean, now you can heat the tents, but those days we couldn't heat the tents. So in a cool year, there was not much difference because we couldn't heat the tents. It wasn't much different in the tent and outside when it was cloudy and cool. So it worked well in a hot year, the membrane integrity and yield. And now here we look at gene expression and also single and multiple genes. So my PhD student, Nicola, she tested it. This is the control. Now, control is almost the same, but you can see the Robisco activase, which is for photosynthesis. After 1 hour, the heat tolerant variety shows a difference between the heat tolerant variety, Cyclop 53, compared to Cicala 45. So there's a big difference when you have after 1 hour in the Robisco activase. And one of my colleagues, Brian Edwell, he did a similar test with rice, wild rice, and he found he used also Robisco activase. So it's quite similar technique, can be used for different crops. And here we looked at global gene expression. So if you look at the heat tolerant variety, you can see that most of the genes are regulated, whereas In a sensitive variety, some are up-regulated and down-regulated. So it's a bit complicated. So you can see, metabolism and other things. There's a lot of things happening down there. So we look at this. This is good for PhD, but not very good for screening. But I'll show you what we're doing as a next step to develop a screening method. So we were lucky. This one was my second PhD student, or not second, maybe third or fourth. He's from Pakistan, Najib Ullah, right? He's now Dr. Najib Ullah, and he's moved over to, I think, in Brunei now. They've engaged him as assistant professor now in Brunei. So we used in this method, actually this is also a collaboration with someone from University of Faisalabad, Dr. Sawa. Dr. Sawa came to visit us and he helped us to do the work with the heat tolerance. I think he's working with you now, Dr. Sawa. So we have a Pakistan variety brought over, CIM 448, and we have the CSIRO variety, Sycot 71 BRF. BRF means, Bourgaert 2, that time is Bourgaert 2, and Flex means the Roundup Ready Flex. And we also had the Lindless Mutant, which was insensitive to ethylene. So we took the plants, we heat them for 7 days at 45 degrees during early reproductive phase. And then we measured ethylene production and also fruit retention, how many fruits dropped off and how many were retained, right? It's a very simple experiment. And we did 2 lots of experiments. One lot we did with waterlogging because, in Australia and maybe Pakistan also, we have either flood or drought. So waterlogging, the top ones, and with Sawa, Dr. Sawa from Pakistan visiting us, we also did high temperature, 45 degrees, okay? So if we look at the left, this is all ethylene production, and to the right is how many of the fruits were there, like the green ones are the control, so the fruits are sitting there, whereas the blue and red is the waterlogging or the heat treatments where the fruit drop off, right? Drop means like dropping off. So you can see, let's look at the top first, waterlogging. When you look at the waterlogging ones, when you have waterlogging, what happens is that they produce ethylene, okay? Whereas the control that don't produce ethylene. So the ethylene, when you produce ethylene, of course, your fruits drop off, whereas the control still maintain the fruits, right? But there's a difference when you have high temperature, okay? When you have high temperature, there's not much ethylene produced, not much at all. And this is very interesting. But the fruits drop off in the, in the high temperature treatments. So it's very interesting that the both stress cause fruits to drop, but one produces the waterlogging one produces ethylene, and the heat one doesn't produce ethylene. So that's a very interesting one. So we did some more graphs. We looked at the relationship, and we found that with waterlogging, ethylene production increases. We have fewer fruits because they drop off. But with high temperature, it was not the case, right? So ethylene can be, there's no relationship between ethylene and number of fruits. So we track them, right? From all the small pin squares to growth, to white, pollinated, to the green balls. And that's the trends we see. So Dr. Najib then found out that it's not to do with ethylene, it's to do with pollen germination and tube development, which we all know now. Now it's no longer secret. Everyone knows about this. Even some of the work I've seen in thesis in Faisalabad also showed this. So you can see that used in Sycot, which is the Australian varieties, the pollen germination is very low, right? And also pollen length. compared to the Pakistani variety. It means your varieties are much better because you have higher temperatures. So we're using Pakistani varieties now for breeding. You can see much more tolerant to high temperature, right? With higher percentage of pollen germination. Okay, so Pakistani variety much better than Australian variety at the moment. So what happens when you have high temperature like 40 degrees compared to the control? You can see sometimes the pollen doesn't, dries up, doesn't dehisce. You can see that it's very common like this as well. And there's all the differences. You can see all when the anthers are quite different. So so this is some work that has been published now. And also electron microscopy, same thing, dried up with 40 degrees and 28 degrees, they are looking good. And the pollen as well, the same thing, mature pollen, 40 degrees, you can see, looking really bad and not the spike. This looks like COVID, but it's not COVID, it's actually pollen. You can see what happens here. The spike's not looking too good, but with this looking intact, and same thing down here. If you're at immature stage, heated, same thing, they look really bad when they are 40 degrees, smaller as well, whereas the control looks not so bad. Okay, so what stage do we heat them at when we want to see changes? Before I go on, we'll just see what happens in the during meiosis. We all know that during meiosis it changes. They sort of divide from the pollen mother cell, the first stage, to the tetrad stage where they become 4, and then they become the microspores down here, right? Actually, they become dyad first, and then the tetrad is 4, and then the small little microspores, and each microspore eventually becomes a pollen grain. So you can see this is what it looks like outside, right? Inside it's like that, but we don't want to always look in a microscope. We want to see what it looks like, when it's a small little, what you call square. So what we do is we heat them at a square stage, early square. This is pollen mother cell, tetrad stage, right? So and then we collect the samples here, and we do all the proteomics, right, to see what's happening. Okay, so that's quite a simple thing that we're doing, and we put them in, sugar solution, look at the germination rate, look at the proteomics, what sort of proteins are coming out of it. And this was not done by Pakistanis. It was one of our colleagues from Iran, so it's a neighbor of Pakistan, and we did this a couple of years ago, and just published last year, the proteomics. So similar thing when you heat it. You can read a lot about proteomics. I won't talk about it in detail, but you can see the flower size is reduced when it's hot. All right. Same thing. The dehiscence is reduced, and the ovules size also reduced, and pollen germination is also reduced when it's high temperature, and pollen tube length also reduced. Right. So it's in that publication there, in the detail. So in the future work, we have a new machine now. It's from, I think, Switzerland, but we're using it for wheat at the moment. I haven't even tried it on cotton yet because we haven't got to that stage, but because our new project is on wheat. So it's an interesting machine. It's using nanotechnology. So it's a flow cytometer, right? So you put it through, you can measure much quicker. The old days we used, we can do it very slowly because we need to use microscope, we need to use proteomics and stuff, but now we can do this very quickly. So the last publication we have, Bakshi, he's actually from Bangladesh, but anyway, we're doing on wheat. We can put it very quickly, and it's an electrical field, right? It puts it through a field, and then you can tell you very quickly. You put it lots of high throughput, right? You can have a lot of lines. Like wheat, we got like 1,000 lines. We can put it through very quickly to tell us what proportion is viable, what is not viable, and what is too small. If they're too small, immature, it'll show up as well. So it actually calculates for you straight away. You don't have to use microscope or anything. Just put it through this machine, tells you how many percent are viable, how many not viable. Okay, so it's very fast machine. And in the future, this is my former student, Warren Connerty, and they're planning to have similar method, identifying the parents, and maybe what they do is maybe looking at gene editing, not putting some traits in early, doing some crossing, looking at genomic selection with looking at environment and phenomics and genomics. And then doing the selection with that. This is the future way of looking at it, and developing cultivars from there, and then maybe doing a bit of more gene editing, and then releasing a cultivars. So this is the new plan that they are planning. That they haven't got there yet, but this is what we are planning in our future breeding for heat tolerant, waterlogging tolerant, and high yielding cotton varieties. So in conclusion, we found that a few ways that can help us to develop high heat tolerance. One is using electron transport rate, membrane integrity, and also now we're further developing also some viability type measurements to improve. And not just using field work, using also laboratory and controlled cabinet experiments to supplement the field experiments. And we're looking also at pollen viability, but pollen is not everything, because what happens is that we all know cotton is not not indeterminate crop. So actually, if you have a heat wave going on, you lose a few bolls, but you will keep producing more flowers. So it's not the only thing. It's one way of screening. Another way is to look at resilience, how we can survive the heat wave and produce more fruit and recover. So when we develop all this, we're looking at a multi-level approach to screen not just pollen, but also for photosynthesis, for recovery of the cotton. So yeah, I'll be happy to answer any questions. And I'll unshare so that we can see each other again.
Yeah. I'm asking the question in chat box if anybody.
Oh, okay. I need to unshare first, then we can see.
The students will ask questions shortly, and they are just joining. The number is going to increase time to time. And so thank you, Professor Than. Thank you very much.
Oh, thank you.
And you shared all the details, all the physiological, morphological, biochemical, and genomics, phenomics, all the tools that could be used to identify the heat tolerant lines and the inheritance pattern of the heat tolerance that is usable. And most of the traits, we are also using these traits, and some we are not using, and hopefully we will be able to access those tools you are using. Once again, thank you very much, Professor Than. So our next speaker is Dr. Alini. So Alini is a senior principal researcher in agriculture organization, Demeter, and the name of organization is Plant Breeding and Genetic Resources Institute in Greece. So she had expertise of agronomy, focusing on plant breeding, crop breeding, evolution of industrial crops, cotton, tomato, hemp as well. Because hemp, this is quite interesting for us, because Alini, it was not allowed in Pakistan to grow the hemp. So hopefully you will tell us something about the hemp, about its fiber traits as well. cotton and tobacco as well. So she has expertise of fiber quality and evolution. So she had a couple of research proposals and focusing on development of edu-types for salinity and for irrigation states. So we are hopeful that she will tell us the techniques usable for the development of new tools and her title is Greek Cotton Sector Towards the Tomorrow Challenges. So over to Dr. Alini. Dr. Alini, please.
Thank you, Professor Azar. Good evening, ladies and gentlemen. I'm glad to take part in this webinar and present you the Greek cotton sector. Okay with the presentation? We are okay?
Yeah. You fixed the issue. Yeah, thank you very much.
Okay. First of all, I want to say that my bachelor is an agronomist from the Aristotle University of Thessaloniki in Greece, and my master and my PhD is in chemistry in the same university, and my thesis title is Evaluation of Physicochemical Properties of Cotton, Proteins and Fibers. I'm now, as Professor Azar said, I'm a researcher in Hellenic Agricultural Organization, which is established in 2011 following the merger of 4 organizations which are involved in agricultural sector activities. Hellenic Agricultural Organization functions as a legal entity of the public sector, supervised and financed more than 51 by the Greek Ministry of Agriculture, Development and Food. It consists of 11 research institutes all around country in the fields of crop, animal, fishery, forestry science and quality control. More specific, my institute aims to the breeding and seed production of cereals, industrial crops, trees, vegetables, medicinal and aromatical plants. And as Professor said, from 2016, we are dealing also with industrial hemp because this cultivation is allowed in Greece only for varieties registered in the European catalogue, and that the content of THC, tetrahydrocannabinol, is lower than 0.2%. And so the farmers start to cultivate it also, this crop. And now all around Europe, there is a trend to use seeds of hemp for food or seed oil and so on. And also in our institute, we work on the improvement of cultural practices, plant protection and precision agriculture. We develop analytical research method, and the national zinc banks belong to us. The Greek zinc bank established in 1981 for collection, regeneration, characterization, evaluation, conservation, and documentation of plant genetic material. It maintains ex situ over 50,000 samples of different plant genetic resource, and these plants are conserved in the form of seeds, which are maintained in cold rooms. Also, there is a collection of 270 different grape varieties maintained in the field. Our campus is in the north part of Greece, in Thessaloniki, and it consists of 20 hectares of experimental fields of the offices, laboratory, greenhouses, and so on. Now, going to the theme of this webinar, cotton. Cotton is a strategic crop for Greece because it represents 50% of the irrigated land of Greece and 10% of the total agricultural land. Greece, as we probably know, is the Mediterranean area, and the farmers have a very good specialization in the cotton farming. Also, cotton is a strategic crop for Greece because Greece cultivated 80% of the European cotton growing area. The other countries of Europe that cultivated cotton is Spain and a small area to Bulgaria. It occupies more than 45,000 farmers and gives jobs to almost 100,000 families. Cotton, ranking number third exporting agricultural product of Greece. The cotton is not cultivated in the island of Greece, but mainly in the mainland, in the Thessaly area, and in the north part of Greece, Macedonia, and Thrace. The last year, 2021, the cultivation area it was 262,000 hectares, and the production of seed coat it was 850,000 tons, while the production of lead is 290,000 tons. Most of this lead is exported, almost 90% of it. Only 10% of the Greek cotton used for domestic use. And the countries that this cotton exported is mainly Turkey, which is the neighbor country of us, and then Egypt and other countries in small proportion. As we can see in these graphics, Greece is a cotton exporting country in the top of the cotton exporting countries. At the year 2020 is third, and in the year 2016, it was 8th in this rank. About the Greek cotton cultivation, Greece is in the verge of cotton cultivation, and the characteristics of the crop is the small, highly specialized cotton farms. The average area for a farmer in Greece is 4.5 hectares. The cultivation period is from April to October, and the sowing was done with machine and 96 centimeter distance between rows. The average plant per hectare is 150,000 to 170,000 plants per hectare, and all this area irrigated with drip or spray irrigation, while the harvesting was done mechanically, and the farmers sell seed cotton to ginneries. The seed that they use the farmer are certified good quality with high germination and disinfected, and has to choose varieties from the European data catalog. This is a catalog of all the varieties, and more specific for the cotton, they are registered 193 varieties, 105 of them from Greece, 55 from Spain, and 30 from Bulgaria, and only 3 from Italy. The research focus about the breeding of varieties focus on high yield varieties with good quality and with reduced inputs. And as I said before, growers with the passing of years gain a very good level of knowledge and experiments in all agronomic practices. The farmers certify their production according to the Quality National Scheme Agro 2, which aims to the control of all stages of the production process, reasonable use of fertilizer, water, and plant protection products, environmental protection, health protection of producers and consumers, production of safe and quality agricultural products, and continuous information and education of cotton value chain. This scheme has developed my Hellenic Agriculture Organization, our organization, and harmonized 2020 year with BCI Better Cotton Initiative Protocol. And so all the production certified with Agro 2 had also BCI certification. The organization dealing with cotton in Greece, except from our institute, as I said before, there are also other institutes, the Institute of Industrial and Forage Crops, and more specific, the National Cotton Classification Center of Karditsa, which is classify all the production of wheat in Greece. Another organization is, of course, the Ministry of Rural Development and Food, which deals with implementation of European cotton agricultural policy, with the strategy development and legislation, the varieties registration and assessment, the list of plant protection products and bioscience, and crops information, which are all available at the website of this of our ministry. Also, there are organizations like OPEKP, which deals with payment and control agency for the community end, and the Greek Interprofessional Association of Cotton. The Greek Interprofessional Association of Cotton, along with Hellenic Cotton Association and Exporters, and various associations of Europe, are the founding members of European Cotton Alliance, which established 2019. the vision of European cotton is to develop a European cotton collective trademark, to acknowledge European cotton as a high-value, sustainable source material, to contribute to branding and promotional strategy, to promote and increase the cotton production in the European Union through sustainable and environmental-friendly practices, to have traceability of European production, to establish the harmonic collaboration and cooperation among the cotton producer and the textile industry, of course. As sustainable production is the theme of today's seminar, I have to notice that in Europe, we focus nowadays on conservation agriculture. According to the FAO, the 3 principles of conservation agriculture is the minimum mechanical and soil disturbance, which means that we have to use reduced tillage system with no inversion of soil layers, to have the soil coverage with the residues of previous crops or cover crops, the biodiversity with crop protection, cover crops and intercrops, reducing the fallow period. In the frame of this, we participate in the CAMA project, which is funding from European Union under the auspices of PRIMA call. More specific, in the frame of conservation agriculture, I will present you some experimentation that took place in our institute, and more specific, the cultivation of cotton with lentil. In this experiment, we cultivated 2 rows of cotton next to 2 rows of lentil, and the next year, in the area of, in the rows of lentil, we cultivated cotton, and in the rows of lentil, lentil. The results show that the cotton yield was sufficient as high as the average in Greece, although the lentil yield was lower than average. With this experiment, we reduced the nitrogen input the second year in the half. The same experiment replied with chickpea in 2 row seeds as the previous experiment, and although the yield of cotton and although the yield of chickpeas was greater than the yield of the average increase, the yield of cotton was low, because we think that the reduction of cotton yield is due to the fact that the maturity time of chickpea is later than the lentil, when the cotton was in the flower stage during July. In order farmers to be familiar with cover crops, we have an experimentation also with winter pea, Pisum sativum, which is used as cover crop in experimentation field of our institute. The cultivation of peas destroyed before the sowing of cotton, either mechanically in the conventional part of the field, or chemically with glyphosate in the field that we use strip tillage and no tillage. The results show no statistical difference in yield between the 3 system used, but we managed to have better wind control. Another project I would like to present you is the project Tomorrows, which is funding from ARPANET 2 Call of Europe, and the aim of the project is to establish crop management ideotypes under simulating current and projected future climate change with comparative studies in Greece and Tunisia. The partners, except from our institute, is the Agronomic Institute of Tunisia and the CIRA, the Organization of Agriculture of France. In the frame of the project, I will talk now about cotton, not with wheat, because the project is also for wheat. We establish experimental fields in 2 countries, Greece and Tunisia, for 4 varieties, 3 Greek varieties and 1 variety from Africa. We applied 2 fertilization levels in these fields. The normal fertilization that usually used by the farmers, and we reduce the fertilization to the half. Also, we use 4 irrigation levels. The normal and the maximum is 400 millimeter of water, and then we reduce it to 55% 50% and 25% of this irrigation. In the frame of this project also, we established remote sensor for soil moisture, temperature, and soil conductivity in the fields in order to correlate these measurements with the meteorological data, agronomic measurements and treatment, and design ideotypes predicting the effect of climate change in cotton for the Mediterranean area. In the field, we took agronomic measurement, synthesis day, number of nodes, leaf area index, span, biomass of leaf, stem, reproductive organs, wet and dry weight, yield, ball number and weight, and of course technological parameters which measure with HVI, length and length uniformity, micronair, strength, elongation, and color. This period, we evaluate the results, which are not published yet. These are the result of the 2 years, but now the first results show to us that the reduction of fertilization to all varieties has no effect neither to measure agronomical nor to main technological characteristics. Irrigation reduction to 50 or 75% of the normal has significant impact in the yield because it reduces the biomass, the height, the node number, and ball weight. But 25% of reduction of irrigation has no effect on agronomical characteristics, affected yield, and no effect on length parameters and micronair. This result that we can reduce the fertilization almost to the half and the irrigation to 25% is of extremely important for Greece because, as we can see from this data of International Cotton Advisory Committee, Greece is above the average of the world for the liters of blue water used for production of 1 kilogram of lead. More specific, in Greece, we use 2,563 liters of blue water in order to produce 1 kilogram of lead, while the average of the world is 1,931 liters. Also, about the fertilizer, the average world is 219 kilogram per hectares. The Greece is below this limit, is 88 kilogram per hectare. But nowadays, when the cost of fertilizer increasing continuously, it's of great importance to reduce the use of fertilizer and irrigation in order to have good yield and no effect to the quality. All this research helps to produce European cotton of high quality with high spinability, large stretchability, of premium cotton seeds that the seeds used in Europe are no GMO. All the Europe cotton is machine picked, contamination free, and the European logistics are fast and reliable. The statement that same day shipment is not an exaggeration for Greece when the same day that the cotton is ginning, the same day can be in the ship and go to the for exportation. Closing my presentation, I would like to mention once again that European cotton produced responsible and according to European directives and regulation of common agricultural policy, produced under sustainable techniques, according to cross-compliance and greening, it's safe for farmers and secure for consumers. It's produced according to national quality certification schemes, and for this reason is eligible for the Ecolabel Award. I want to thank you very much for your attention, and this is my email if you want to have a contact, or you can ask me whatever you want now.
Okay, thank you very much, Dr. Eleni, for your comprehensive presentation about the cotton, how it is managed, how it is grown, and how the agronomy has been fixed in Greece. And you also told about the genetic resources available with you, and hopefully, but we know all that is non-GMOs. So hopefully those will be usable for us in breeding programs as well. and for the development of some mechanically suitable cotton lines. Thank you very much, Dr. Alini. So
Thank you
our next, soon we'll be in touch with you, and my students, they will be in touch with you. Okay. Now our next and last speaker is Professor Manzoor Ahmed. So he is a leading cotton breeder in Pakistan, and he developed several cotton lines, and most of them, they are suitable for hot areas of Pakistan. So based on his efforts and based on his contribution in cotton, the government of Pakistan has awarded a civil award recently to Professor Dr. Manzoor Ahmed. So over to Professor Manzoor Ahmed for your presentation, please, sir. Sir, please start.
Are you listening, please be?
Yes, sir. Yes, sir.
Okay, okay, thank you. First of all, I welcome Dr. Tan and Alini for this joining this webinar, joining excellent event organized by Dr. Tasir. And I have firstly 1-2 question for Alini, but at the last I will ask some question for my information. And my today's talk will be covering the current challenges in cotton production in Pakistan and the NAB Institute where I'm working, what we are doing in developing our climate resilient cultivars in Pakistan. Present stop. Okay, what happened? Okay. Are you listening, please?
Yes, yes.
Oh, okay, okay. This is about the brief about the background information. The Pakistan economy can be rightly called as mono-commodity economy because of its exceptional dependence on cotton.
Sir,
Considering its,
please share your PPT, sir. We are unable to see.
You are unable to see?
Yeah, please share your PPT.
Oh, okay, okay. Okay.
Yes, it's fine. Okay. Please go ahead, please.
You see my first slide or not? Okay.
Fine, sir.
Okay, okay. Thank you. The current challenges in cotton production in Pakistan. This is my today's topic, and what NAB is doing in developing of climate resilient cultivars. I will share all about these information. About Pakistan, when we say about its importance in cotton, about 55% contribution it's in foreign exchange earning we get from cotton. And it's a 1.6% GDP and 7.8% in total value added in agriculture. This leading natural fiber crop in Pakistan is grown on 3.2 million hectare with production of 11 to 13 million bales. And if we in the Asia Pacific region and consider China, India, Pakistan, we are contributing with this Asia Pacific region is contributing more than 60 to 65% in total world cotton production. Historically, good things have happened for the improvement of cotton in Pakistan, but the erratic trend of cotton production in Pakistan since 1992, after the appearance of once nuisance disease of cotton leaf caused virus disease, our production was in debacling phase. Like Pakistan, cotton is also playing very important role in the economies of other countries like Australia, China, India, Iran, Myanmar, Vietnam, and Bangladesh. If we compare our yield in with other countries, we are still our production is 700 kg per hectare if we compare with China, India, and other countries like Australia, we are still behind. The yield difference, these yield differences are amongst regional countries are being aggravated due to changing climatic condition like high temperature and unpredictable fluctuation in the rainfall pattern. Pakistan is likely the country that is being most affected from these climate changes as far as the agriculture and cotton production is concerned. As in the projected temperature increase 2.7 and 3.2 under these RCP, respectively by the mid-century, along with 18 to 30% in rainfall in Pakistan. This is the AgMap data of 2016. Firstly, we observed is the disaster effects of heat stress were dominant Pakistan during the year 2014, where our early termination of crop with the 40 to 50% of abortion of fruit. The cotton belt of Pakistan is mainly located in the region of high temperature with maximum temperature often exceed 45 degrees centigrade. But the optimum temperature for successful cultivation is required is 35 degrees centigrade. I will just have a mention here about the flare of cotton, and the 2000s flare of cotton started from the year 2015-16, where 34% shortfall in total production with the alarming demand of 2.9 billion bales by textile export started in the year 2016. And much of this variability and shortfall in yield has been attributed by high temperature and heavy rains. We in Pakistan, we are facing both issue with the high temperature, flare of the insect complex, and with the heavy rains, with the early termination of crop, and so many unable to the growth stops, and the farmers are unable to get the high more yield, and failure their crop often failure. And this year-to-year variation yield of cotton crop in Pakistan has become a major concern of cotton producers and other stakeholders related to cotton-based industry. Taking the year, this is the review. In the 2015-16, 34% shortfall in India, in China, and in the world scenario. But the year, the challenges, just I mentioned, we have the early challenges, the current challenges, and the most the alarming challenges basically is the new climate change, new issue of climate change. What happened when we shifted to this our regime of the GMO cotton in Pakistan, and our traditional varieties, as Tan told, that were very tolerant to high temperature stress. The GMO variety failed to cope with the enhanced attack of whitefly that increased due to high temperature and shifting. That's why the farmers shifted to alternative crops, decline in traditional growing area, increased pest control cost in spite or because of BT cotton used with ultimate less return from cotton. And what we faced alarmingly, the pink bollworm threat and whitefly became out of control. Okay. Here is you see just I do what happened. The core issue became the whitefly issue, sleepless night of the cotton growers. All this happened due to the succulency sensitive GMO cotton against heat stress sucking complex, and farmers were very poor, very poor awareness of growers about this varietal response against the sucker complex. These are the causes, and what happened due to this GMO crop, high number of sprays, ETL level were disturbed. Question about the efficacy of products raised, high cost of production, and farmers decrease income farmers' income and yield per unit area, decreased supply of raw material to the industry, farmers shifted to the alternate income. These were the effects, and all what happened due to this climate change and the our varieties, the previous variety which were variety grown as GM failed to cope with the changing climatic issue that we face. How does high temperature affect the cotton? You are listening, please?
Yes, sir. Yeah. Please go ahead, please, sir.
Okay.
Yeah.
How does cotton plant? How does high temperature affect the cotton plant? Increase trend of temperature inhibits the growth of plant with ultimate impact on the increase photorespiration, enhanced attack of sucking complex, and poor control for insect pests. Here I would like to mention here that in Pakistan particularly, we are facing a lot of sucking pest complex, particularly with the increase of high temperature. The white fly, just I mentioned, becomes out of control. Or with this, the enhanced requirements for inputs like fertilizer, higher cost of production. Here I try to how the impact of increase temperature affects is the total fruiting sites, intact fruits, and fruit shedding. This shows the fruit shedding, the total fruiting position is decreasing, but the intact fruits is decreasing due to high temperature with the increase of temperature trend. Cotton production under the changing climate scenario and strategy to cope. Year 2050 big issue. Here I mentioned the high rains plus high temperature was considered the main cause of disaster. But taking the year 2060-70, big issue of sucking complex, particularly white fly and pink bollworm, became out of control. Here the normal rains, but was the high temperature factor. Flare of white fly main cause of disaster, and the all happened. Interestingly, the varieties we shared with our the mutant varieties proved as the climate resilient, and that proved as the more laterally you will see how these variety adapted to help to cope with the issue of climate change. Here are I would like just here to show the year. This is the year 2012-13 when how the high temperature were affecting, heavy rainfall affecting this. You see, I tried to show the comparison. This was the year 2012-13. This is the season of the heavy rains when you see the top of the crop became barren. But on the other hand, you see after heavy rains, even after heavy rains, there is no shedding of fruits because you can see this is due to this pollen sensitivity, and you see the resiliency of variety against the heavy rains. And this is the year you see I have tried to show here the year 2014-15. You see this is the control variety where just the GMO variety, and you see the lower half of the is totally barren due to shedding of high temperature. On the other hand, you see our mutant variety, heat tolerant varieties with the retention of food at very early, and there is no gap, and there is continuation in fruiting with this bole size, in opening and traction with fluffy bole size, and as so many parameters. Just to show here the year 2000 taken the year of 2018-19. How does this dense foliage GMO varieties help in the flare up the fee. So this is all due to blackening of the cotton of the lower portion, and this is the field view under the, you see flare up of the white fly type, you see, and how the crop is being damaged by the high temperature and high. Interestingly, this is I will try to show that this is how the cotton butterline. This is our non-GMO variety. This is the, and this variety was approved in 2013, and this is its name is Neopiran variety, and this is the other GMO variety, and you see how the succulency of the leaf GMO variety is unable to tolerate the pressure sucking complex. On the other hand, you see the tolerance of the variety of against the sucking complex with with this potential continuity and fruiting up to top with the UCF. You see there is a discontinuity and there's a continuity and a fruiting. Under the field condition, if I to show the how the heat tolerance under field condition, you see the heat sensitivity of the material, and this is the due to polar abortions, shedding or abortion of fruit. On the other hand, you see no shedding of fruit in the field, and this is heat tolerant variety. Okay, just okay. Now I would like to share the success story on induced mutation for the improvement of cotton in Pakistan. We started the pioneer work on through induced mutation in early 70s with the objectives to develop cotton variety with early maturing coupled with enhanced yielding ability, wide adaptation potential, and resistance to biotic and abiotic stresses. However, NIAB 78 cotton variety was first landmark achievement in the through pioneer work through induced mutation that revolutionized cotton production from 2.9 million bales to 7.2 million bales. And about it covered with this variety covered about 80% of variety and the Sindh. And if we say this variety proved as the longest life variety in the history of cotton in Pakistan. What had helped the industry, early arrival of seed cotton in the genic factories, and brought great evolution prosperity among the farming community, and with the increase in yield, the cumulative impact have been with the its to the country's economy. The overall objective of the development and evolution of cotton variety having the ability to unlock the yield potential under the changing climate scenario, high temperature, high rainfall, with particularly focusing with the lodging resistant, heat tolerant, deep root system with the ability to stand under the moisture stress both under high and low, and high yielding ability with their wider adaptation potential with the fine quality fiber traits. Achievement rate through what we improved through the induced mutation. You just to share the awareness rate with the you see the retention of bowls at wet window. These particularly leaf orientation, facilitating the maximum penetration and penetration of solar radiation, and its particularly is petiole length, leaf is petiole length, it's supporting this trait. The number of varieties we develop, particularly the starting from NIAB 78 and the year from 2013-16. Just I mentioned these variety 2013-16 and this variety in 2017. In 2 years, I mentioned 2 years Firstly, this was approved in one province. Considering its wide adaptation potential, this variety was approved in another province. And the same with this variety. And NIABS 878. Currently, these both are the leading varieties in the country. And these are the recently approved varieties. And now with big contribution in seed production system. Reason of gaining popularity by India varieties among the farming community due to their heat tolerant with ability or retention of fruit very low, medium small leaf with stay green character, easy control of insect pests with maximum penetration of applied insecticide, lodging tolerant and resistant, ability to sustain their yield potential under the changing climatic scenario. Here I would like to mention here that when we are working for heat tolerance, we go for to evaluate to the material to be evaluated by the independent by the independent researchers like plant physiologists. We share from this you see the data I shared here. You see the pollen fully disintegrated pollen viability and relative cell injury levels. This week is country there were so many other varieties. The higher values with less tolerant relative cell injury values. The NIAB 878, the most currently the most heat tolerant variety grown in Pakistan. We continue study for 2 years this variety for its heat tolerance and its value was relatively its cell injury was 39 and with the pollen viable above 90% and the full day center and above 80%. And for the next year to the 15-16, this again is anthrodistant value 93, pollen viability 97% with the relative cell injury 90 39. All these traits favor as supported us in deciding that not only this but in the under field condition this variety not only performed excellent under the heat stress area, totally heat stress prone area, but also replaced the other varieties previously grown. Here I would like to just share the current updates about NIAB cotton varieties grown in Pakistan. Yes. I mentioned currently we have the about taking the one public sector organization. Currently, our variety have 87% share in the seed production share, and considering the heat tolerance and wide adaptation potential. This from the multiplication targets with this our public sector organization. You see our share. This NIAB varieties more than 50% in the seed production share, and in the year 2020 our share rose up to 59%, and at 2018-19 how gradually we move towards higher side. The field performance of by this is the to harvest good yield G into E into M, and this is our main cotton growing area like Melsi 2017. This is the performance of its NIAB and being visited in Melsi area. You see how it's this variety is performing the field. You see its leaf area, its retention at very early node, and no gap in fruit shedding, and you see continuous fruiting. The material here I would like to share that accordingly the varieties which we need under the this changing climate scenario, we need the varieties which are early and indeterminate growth habit. We develop those varieties, and that's why we are successful, and we are try to inshallah hopefully we are trying to hope to help in reviving of the cotton production in the country. You see, and from this you see how there's a leaf area, how the is performed by medium small leaf area, and very less attraction in the sucking to against the sucking complex, and can tolerate. You see, trying to visiting this the Sindh province, the also the heat prone area. You see more than it's from its plant field. You see its picking is more than about bowl opening is above 80% a bowl opening in the field, and with the you say bowl opening in the field, and still is a green character, stay green character. Looking for the, just I mentioned that these are the traditional varieties like GM variety with you see GM with the scarcity of fruiting. You see the our mutant line continuous and with perfuse bearing, and we are looking for these type. We are working on these type of varieties. Here I would like to mention that the year was 2020 worst year for our for cotton in Pakistan. You see here this is the you see lower half of the crop affected. There are heavy rains, particularly for 1 week, and normally the varieties start growth their growth terminates. But interestingly, I will like share our the mutant varieties did not stop. You see, although the lower half ball factor, but it recovered, and its fruiting continue with no gap, and no fruiting is stop of fruiting, no and growth, no termination of growth. And this is the Niab Kiran variety. You see it's again you profusely bearing at the top, and the also the same trend with the bole bearing retention at very small leaf area, medium small leaf area, and this is another example of the Niab 545, another variety year approved in 2018. You see also retentionable at variety area 6-7 node we our variety started, and again interestingly you see the top with the bearing top with excellent bole bearing, and that's why we need these variety type of variety that can that may be early and indeterminate growth effort. And you see again it's more another field view. You top with bole size with bole opening, and this is another variety, and you see this is Niab 1048. This is excellent. You see bole opening as attractive, and the you see its top with the good bole size from bole starting for 6/7 node again. These varieties, and no retardation in growth, growth is not affected. And this is newly approved varieties, heat tolerant, dosing resistant with duper system. And this is a newly approved variety just this year. This we propose the its name is Niab Sanabim, Save Agriculture through nuclear application and biology, and this is for mutation. And really this variety has also performed excellent this year, and approved by the government of Pakistan. And you see it's again its potential retentionable at very early with continuous fruiting, bole size, and the fiber is very fine, above 31 millimeter length, with fineness is 4.1, 4.2 in the range of cotton being visited by our official, by government. And here I can show, if you see some, yes, is this can be translated. I will also translate a few words. This is the our one grower in the Leia, another cotton grow area, that our totally sandy soil. And how this variety is being performing here is telling that under the very poor soil, this NAB 1011 is performing excellent, considering its earliness, its ball-bearing habit, its first pick, what expected in the next pick, and this field is selected for the comparative for comparison for the announcement of prize for farmer. The best winner for the best variety they selected the farmer. But they are expecting that the ready bowls and the open bowls and the what they expect. They say that above in more than 14K, 1400 they are expected. And they are telling about that its leaf size, considering its leaf size, very less attack by white fly and very less attack of pink bollworm, and that's easy to manage this. The our provincial minister visiting this year to our cotton experimental areas, and you see the field here, see the potential in our with the mutants we have, and this is our provincial minister, and this is the special task force. The briefing is given to the special assistant to prime minister, and about the performance of our cotton mutant. These are other high dignity from my country. A briefing is given about the performance and impact of the cotton variety, what we develop, how our variety develop are contributing the impact. And interestingly, this year, what I visited in the field area, you see this is a variety, you see abortion of fruit at the top. This is another variety that in the country, its behavior and indeterminate growth habit. Also, when you plant this type of variety, if there any stress, unable to, this variety is unable to cope with any kind of stress and terminates. And farmers, that's why it's a growth stop in the month of by the end of August, and farmers are unable to get more yield from this variety. And the how now the just to close a way forward to look for review of cotton variety that can sustain their yield under the changing climatic scenario, and with high tolerance against high temperature and sucking compress, lodging resistant, deep root system with ability to withstand under moisture stress condition high and low. Here I say what high and low. Low moisture means they can survive under the drought condition. When I say they say about the highest moisture, heavy rains. When the heavy rain, they can tolerate the waterlogging stress, and that's why this will be only happen when your deep root system is supporting your material, your system. And looking for the build high yielding variety, need to for strict regulation. These are other things in my country, strict and justice. Another thing what we need currently to know, we are trying to restore the confidence of farmers by making cotton as profitable, because if our farmers are disheartened, hopefully this year our area gains is being revived. Inshallah, hopefully this year we will achieve our targets of sowing, and hopefully we will start to our revival. And only this is due to this only we can happen if we have the varieties which can because main role is the variety of management also. But if you have the variety in your management, and if any or variety, if you have the good variety, you have it can tolerate any kind of stress. You can be successful in achieving best yield. And hopefully with these things I will try to my finish talk. Thanks everyone.
Yeah, thank you very much, sir. Thank you very much for your comprehensive presentation about the scenario of cotton. You presented the landmarks, achievements, and the future aspects, what you are going to do and what you are thinking about the cotton in Pakistan. And we are hopeful that cotton will be revived soon, because Pakistan, for the first time, government of Pakistan, it fixed the support price for the cotton grower. So at the end, I'm thankful to our speakers, Professor Daniel Khan, Dr. Alini, Dr. Manzoor Ahmed, and my colleagues from the Department of Plant Breeding Genetics. And we have listened the different scenario of cotton crop throughout in 3 regions, in the European region, in Pakistan region, and Australian region. And all they are affected by the particularly high temperature, which is going to increase every year. And if I say this time in Pakistan, temperature, it exceeds 48 degrees Celsius
Yes.
during the sowing of cotton. And farmers, they faced a lot of problems, and the germination was very poor. And hopefully, from these talks, from our presenters, we'll be able to design the strategies, we'll be able to design the varieties suitable for the farmer community. And we are very hopeful that our V3 partners will be able to design the research proposals we'll be able to exchange some ideas, and we'll be able to place our students for getting the techniques usable for the development of such lines. So thank you very much, all of you. And I'm very much thankful to my colleagues from the Department of Plant Breeding Genetics, our postgraduate students, undergraduate students. And I want to mention here some of friends from Punjab Agriculture University, Ludhiana. Dharmender Pathak was there. Professor Mani, Dr. Mani Vanan from Cotton Research Institute, Combidor. They were here, and they sent me message in private, in a WhatsApp, and they enjoyed these presentations. And hopefully, I'm unable to read some of their names, like Harjeet Singh is there. He is also a professor in Punjab Agriculture. So hopefully, we will design another webinar by having some ideas from Indian side, because they're also the effects of high temperature. And they're also having same issues like Pakistan, having the issues of whitefly, having the issue of pink polvorum and high temperature, water shortage like that. So once again, thank you very much. And we are having like 50 plus students are there. So once again, thank you very much. And now I'm going to close this webinar. Thank you and take care.